Okay, so on to the next little part of gene technology. So in the first little part of it, we learned how small amounts of DNA could be amplified, and that was using the polymerase chain reaction, or PCR. We learned how specific genes could be detected on chromosomes, which was using DNA probes. We also learned how specific nucleotide sequences could be detected, which was using probes as well. So in both of these examples, probes were short sections of DNA which were single-stranded and had a known sequence. So we'd normally create them to be complementary to a section of DNA which we knew caused a disease. We also learned how enzymes could cut DNA at different recognition sites. So this was restriction endonucleases. And then we learned how genetic profiles could be produced for individuals. So this was genetic fingerprinting, and this involved lots of things. So it involved using restriction endonucleases to chop up their DNA. It then involved us using gel electrophoresis, because once we chopped up their DNA using the restriction enzymes, we would all have fragments of different lengths, so we could use gel electrophoresis to sort them by size. We could then use DNA probes and we could label them, and then we would get the genetic fingerprint produced at the end. Okay, so just in case there's any confusion, gel electrophoresis is where the DNA fragments are sorted by size, and genetic fingerprint is the thing that we get left over at the very end. So what we're looking at in this section is what happens after the desired section of DNA has been identified and isolated. And the whole thing about this is basically using little sections of DNA and insert them into bacterial plasmids. Okay, so the main focus of this topic is how we can genetically modify bacteria to produce human products. So the most common example is using bacteria to produce insulin, and that's what we're going to focus mostly on. So basically, what we need to do first of all is get the desired section of DNA and isolate it. Now, we did talk in this about how, or in the previous topic, how we could get the messenger RNA and we could isolate it from a cell where it's likely to be. We could use reverse transcriptase, and then we could use DNA polymerase. So this is what we're talking about here. Now, the most common example of this is inserting the human insulin gene into bacterial cells. However, the gene is not simply inserted into the bacterial cells. There are some important steps involving some enzymes. So these are the three main enzymes. Restriction endonucleases, as we talked about quite a bit, these cut the DNA into pieces or fragments, is how we described them previously, by attacking base sequences in the polynucleotide chain. So this is where we talked about the recognition sites. The restriction enzymes will recognize specific base sequences and cut them at a specific part. Then what's really important in this process and this topic especially is that we get the sticky ends, which we know are the exposed bases. We then have DNA ligase, which can join together lengths of DNA. And we have reverse transcriptase, which can be used to synthesize lengths of copy DNA from messenger RNA. So that's what we've been talking about up in this section. So this arrow is basically represented by the role of reverse transcriptase. Now, the discovery of these enzymes made it possible to remove portions of DNA from one organism using restriction endonuclease enzymes, and they can then be incorporated into the DNA of a different organism using DNA ligase. The new DNA is called recombinant DNA, and for that reason, genetic engineering is sometimes known as recombinant DNA technology. So it's basically where we're combining the DNA of two organisms. Now, just a wee quick recap. So we talked about restriction endonucleases, and this basically shows two types of them. Ones which produce sticky ends and one which produce blunt ends. So for these restriction endonucleases, they would recognize a particular sequence. So for the EcoR1, it would recognize the sequence GAATTC, and it would make a staggered cut between the G and the C. It would break the hydrogen bonds in this area. And that would leave us with exposed bases, which is where the phrase sticky ends come from. The reason why we say these are sticky is because they could form new bonds as long as we match up a piece of DNA which has complementary bases to it. On the other hand, other types of restriction endonucleases produce blunt ends. These ones are less useful for this recombinant DNA technology. Now, we've also looked at reverse transcriptase. So we basically said that if we start off with a copy of messenger RNA, and remember, messenger RNA bases are A, U, G, and C. So if we had A, U, G, C, A, U, and this is a piece of messenger RNA, well then what we could do is we could use reverse transcriptase and it would produce a piece of complementary or copy DNA, which was single-stranded. So because we're going from RNA to DNA, the bases are going to be A, T, G, and C. So the complementary base for this T is complementary base for this A is T, then an A, C, G, T, A. And this is our single-stranded piece of complementary or cDNA. We could then use DNA polymerase to make it double-stranded. DNA ligase is another enzyme that we use. This is simply used to join the two sticky ends of two separate pieces of DNA together. Since it joins the two strands of DNA together, it forms phosphodiester bonds. Now we'll remember that DNA has this sort of ladder shape. The long parts, or the vertical parts, is the sugar phosphate backbone, and the sugar phosphate backbone is basically repeats of phosphate followed by sugar followed by phosphate followed by a sugar that's not a very good pentagon, and then phosphate, and that just keeps repeating and repeating and repeating. Okay, and then we would find the bases in there, and those bases would join together with the other sides of the DNA strand. So these bonds in here are called phosphodiester bonds, and those are the bonds that form whenever the sticky ends are joined together. Okay, so this here, little bond there between that A and the C that you can see really carefully in that diagram, is a phosphodiester bond. And then these little ones in there between the T's and the A's and all that sort of stuff, they are hydrogen bonds. So basically what we've got here is we have got a little bacterial plasmid. Now we'll talk about this in a wee bit more detail later on. So there's a bacterial plasmid, there's a human gene for something. Both of them have got sticky ends. And if you look carefully at the diagram, you'll see that up here we've got TTAA, which is the exposed bases here, and here we've got AATT. So we should be able to join those together. So when we join them together, DNA ligase basically sticks them, forms these new phosphodiester bonds, and we've got a completed plasmid. Now, before we get into the next wee part, gene transfer, and before we sort of start talking about this process, we have got a few very, very important words, okay? So basically, recombinant DNA technology. This involves us transferring DNA from human DNA to bacterial DNA. So, a couple of key words in this that we're going to use: donor, host, and vector. Okay? Now, we still use all of these enzymes and all that sort of stuff, but these are some key words, right? So, the donor is the organism whose DNA is to be transferred. So, in the genetic engineering and the production of insulin, the donor would be the human. The host is the organism into which the DNA will be implanted. So in this case, it's going to be the bacteria. And then we've got a vector, and the vector is a plasmid from a bacterium or virus. Now, I suppose before we go any further, we'll probably need a little recap of a bacterial or a bacterial cell. Okay, so here we have the little bacterial cell. We've got a cytoplasm, we've got a cell wall, and then we've got a plasmid. Okay, now the plasmid is how the bacteria stores their DNA. So you might remember from GCSE that a bacterium has no nucleus. So the plasmid is where it stores its DNA. So all its genetic information is stored in this little loop of DNA. So what we want to do here is try and transfer the DNA from the human into the bacterium, and then if we've transplanted the correct gene and everything's functioning correctly, we should be able to make the bacteria produce a human chemical. So once the desired gene has been identified and isolated from the donor, so this is using all the techniques that we've described. So for example, using PCR to amplify a small section, using the reverse transcriptase, all that sort of stuff. Once we've isolated from the donor, it's transferred into the vector. The vector is then used to transfer the donor DNA into the host or target cells. Now, let's try and apply those to this little diagram here. Okay, so we've got the plasmid DNA, which we know is the vector. So it's basically like a transport medium. It's how we can transport one piece of DNA into another organism. We've got the human cell, which is the donor, and we've got the bacterial cell, which is going to be the host. So what we can see here is that if this is the human cell with its DNA in the nucleus, we can extract the DNA, take out the gene for insulin, and then we can implant it into the bacterial cell. Now you'll notice here in this section that the plasmid gets removed and then it gets cut open. So this would be cut open using restriction endonucleases. This DNA would also be cut using restriction endonucleases, and it's important here to say that that would be the same restriction endonucleases which cut out the human gene and cuts out the bacterial DNA. The reason why that's important is because if we use the same enzyme, they'll have complementary sticky ends, and it'll make it much easier to join it up. When we get the human gene and the cut plasmid, we just use DNA ligase to join them together. Then what we're going to do is implant this into the cell. And hopefully what should happen is the bacterial cell should reproduce and we should start to produce loads and loads of product. So there's loads of steps that we're about to look at here. Okay, first of all, we need to look at how restriction enzymes are used here and here, and then we need to talk about how ligase is used there, and then what we want to do is talk about how we can actually check to make sure that this is the correct bacterial cell that's been produced and that will actually work and it will actually produce the product that we want. So there's quite a few wee steps to look at here. So inserting the gene into the vector. As we've said before, the vector are plasmids and viruses. Now, plasmids are the ones that we talk about most. The plasmid is the closed loop of DNA, so it must be cut open to create ends to which the donor gene can be joined. Now, luckily we know that we can create ends using restriction enzymes, and these ends that we're going to get are sticky ends. So we take our plasmid and we've got to cut it open. We want to use the same restriction enzyme to cut the plasmid and cut the donor gene. So this means that the plasmid and the donor DNA will have complementary sticky ends. So the donor DNA will join perfectly with each end of the cut plasmid, and that's because they'll have complementary exposed bases. The DNA ligase is then used to anneal which is basically a fancy word for join the DNA backbones together through the formation of phosphodiester bonds. And these bonds were between the sugar and phosphate molecules in the backbone. The end result is that the donor DNA is spliced into the plasmid, which becomes a closed loop again. And the plasmid DNA is now called recombinant DNA. So this part here is our recombinant DNA. And we would say that the bacterial cell has been transformed. Now, viruses, these are naturally adapted. Sorry, I should say, right, we did say at the start that the main types of vectors we can use are plasmids and viruses. Plasmids is the most common one, but viruses can also be modified. Now, the thing about viruses is they're naturally adapted to inject their genetic material into a host cell. A bacteriophage could be treated to have donor DNA spliced into its own DNA, which basically means that the donor DNA will also be fired into the host cell along with the viral DNA. So basically what we need to know about this is that viruses are naturally adapted to inject their genetic material into another cell, and that's basically how they spread. So what we can do is we could take the virus, we can modify it, insert the human gene into a virus, and then when we release it to come into contact with certain types of cells, they will automatically inject the gene that we want the other cells to have. So it's quite clever, but by far plasmids is the most common route that we're going to be looking at. Now, what we need to do then is, once we've got to this stage, let me just get rid of all this writing. So once we've got to this stage, which is where we have inserted the gene into the vector, or inserted the gene into the plasmid, we want to try and get this into the bacterial cell. Now, to be able to do this, we have to encourage the bacterial cell to take it up. Now, it's not like encouraging it, like having a wee chat with it, seeing if you can get it on board and seeing if you can make it do its job. What we've got to do is basically treat it with lots of heat and ions. Okay, so once we have the vector with the gene, the host cell, which is the bacteria, has to be encouraged to take up the plasmid. Now, the thing about this is, what we need to do is we need to incubate them with lots of calcium ions, and we need to subject them to heat shock. So basically what happens is we increase the temperature from 0 to 40 degrees. When we incubate the bacteria with calcium ions and subject them to heat shock, it makes their membranes more permeable, and as a result makes it more likely to take up the recombinant DNA. If they do take up the recombinant DNA, they are said to be transformed. Now, even though this treatment usually works quite well, not all of the bacteria will be transformed. So the next step that we've got to do is work out which ones are transformed and should be reproduced, and those which have not been transformed and so should be destroyed or discarded. So just to recap on that, we have taken the human cell with the DNA, we've isolated it. So we've isolated the gene and cut with restriction endonucleases. We've got our bacterial cell, which is the host cell, and we have removed its plasmid and used restriction endonucleases. We've joined them together with ligase to produce this plasmid, which we said was recombinant DNA. We then want to try and encourage the bacteria to take in the new vector, which is the plasmid with the DNA. So what we've got to do is heat shock and calcium ions. So incubate them with calcium ions and heat shock, and that should allow bacterial membrane become more permeable and take up the transformed DNA, or sorry, the recombinant DNA. If it takes it up, we say it's transformed. And the next step that we need to do is we need to check that everything has worked for us, okay? Now, the reason why we need to check is just because we ply these bacteria with loads of calcium ions and heat shock, they won't all take them up. So basically what can happen is we can be left with three scenarios for the bacteria here. We can be left with a bacterium with no plasmid, simply just because it hasn't taken up a new one. We could be left with a bacterium which has the original plasmid, or we could be left with a bacterium which has taken up the new plasmid, which we'll draw for this one. So this one would be transformed. Now, out of these three, we don't want any with no plasmids because they'll not produce anything for us. We don't want anything with the original plasmid because they'll just reproduce and use up all the resources. So we don't want these ones. Plus they won't even produce the thing that we've been after. So we want to work out a way that we can get the transformed bacteria, because these are the ones that are going to start producing the gene, or start producing the polypeptide that we've been after. So how do we do that? Now, I have to say, this is going to seem like it's absolutely crazy, but this is absolutely genius how they've worked this out. So how do we identify the transformed bacterial cells? Basically, this relies on there being marker genes. Now, marker genes are basically a little section of DNA which tells you something about this plasmid or this bacteria. Bacterial plasmids almost always contain antibiotic resistance genes. So basically, they'll have little genes which make them resistant to antibiotics, which means that they have got genes which make them resistant to antibiotics, i.e. antibiotics won't kill them. So, if the antibiotics won't kill them, we can actually use that to our advantage to work out which bacteria are transformed and which ones aren't. So, if we select a plasmid at the start which has the genes for resistance for tetracycline and ampicillin, which are two types of antibiotics, we can use this to our advantage. What we'll do is we'll use a restriction enzyme which will cut the plasmid within one of these antibiotic resistance genes. So for example, we'll cut the tetracycline gene. Next up, we insert the DNA into the middle of the tetracycline gene. Now let's show you what that would look like. So here we have got the plasmid, and in this plasmid we've got a gene for ampicillin resistance and we've got a gene for tetracycline resistance. So the genes in this part is the bit with the lines, and the gene in this part is the shaded part. So what we do is use a restriction enzyme to cut the plasmid open, and you'll notice here that we're cutting the tetracycline resistant gene. Now, we've also got here the human DNA, and we would cut this out using the same restriction enzyme, and that's so that we've got complementary sticky ends. So we can see here this little section shows sticky ends on the ends of the human gene. We mix them together and we've inserted the human insulin gene into the middle of the tetracycline gene. Now you'll notice we haven't done anything to the ampicillin gene. So what this means is that in this little plasmid, the ampicillin gene hasn't been touched, so it still works. The tetracycline gene has been cut, so it no longer works. What we do then is we'll take this recombinant DNA and we will join it together with the bacteria, or we'll insert it into the bacteria. So this is where we incubate it with the calcium ions and subject it to heat shock, and we should have the recombinant DNA inserted into the bacterium, and we will now have the transformed bacterium. So if we carry on with this, if the foreign DNA is successfully inserted, the tetracycline resistance gene will no longer function. And that's because it's been cut. If we cut a gene, it'll no longer work. The transformed plasmid will contain the active ampicillin gene, but an inactive tetracycline gene. So after we subject them to heat shock and all that sort of stuff, we want to know which ones work and which ones don't work. So those that take up the recombinant plasmid are resistant to ampicillin but not to tetracycline, and those are the ones that we want. Okay, so this basically means that ampicillin won't kill it, but tetracycline will kill it, and those are the ones that we want. Those that fail to take up the plasmids are killed by both ampicillin and tetracycline, and that's because they have got no genes for resistance. So we don't want those. We want to try and kill those ones. But those that take up the original unmodified plasmid are resistant to both, i.e. ampicillin and tetracycline won't kill them. So to work out this part and how we actually work out which ones have been transformed, we basically need to subject them to ampicillin and tetracycline. And to do that, we have to carry out a process called replica plating. Now, if we have a little look at this, this is how it's done. So the bacteria are cultured on agar plates, which we'll remember is a little jelly which is inserted into a petri dish, and it contains all the nutrients for the bacteria to grow. So if the bacteria are placed on the agar plate in a petri dish, the bacteria will multiply to form a colony, which is basically just a little cluster of bacteria. The colonies are then replica plated onto a plate containing ampicillin and a plate containing tetracycline. This involves blotting the original plate with a pad and dabbing this against the surface of a fresh plate so that cells from each colony are transferred. The bacteria that survive on the ampicillin plate only are the transformed bacteria. So if we look at this entire process, right, we start off with the bacterial plasmid. It has got a gene for resistance to tetracycline and a gene for resistance for ampicillin. We cut out the human gene and we cut the plasmid using the same enzyme. We insert the human gene into the tetracycline. So now tetracycline doesn't work. The bacteria are encouraged to take up the recombinant plasmids using calcium ions and heat shock. And then they're placed on an agar plate with ampicillin, and the colonies are allowed to develop. Now, ampicillin, by making this agar plate ampicillin, It basically means that any bacteria which contain a plasmid will be able to survive. So, this means here that colonies only develop from bacterial cells that are resistant to ampicillin, so they must have taken up the plasmids. So any of these ones which don't have a plasmid will be killed straight away. So we know that for definite, any of these little splotches here, they're little bacterial colonies. We know that they will definitely contain a plasmid. But we still don't know, is the plasmid this one, the original one, or this one, the transformed one? So what we do next is we take this agar plate and we make a replica. So they literally get a piece of filter paper, dab it down over the top of this, and then dab it onto another agar plate. When they put the filter paper on it, it'll transfer just a couple of bacterial cells from each colony onto the filter paper. So when they put the filter paper back down on the new plate, it'll transfer those bacterial cells to the new plate. This one is exactly the same. So this has got ampicillin. So again, any of the bacteria here will definitely have a plasmid. But this is the key, okay? They take the filter paper, they dab it on, and they make a replica plate on an agar plate which has tetracycline. Now, this is what's clever about it. Tetracycline in this part was inactive because they've cut the tetracycline gene. So this means here that if we're missing any colonies, like this one, this one, this one, and this one, they can't survive in tetracycline. So this means that they've taken up the transformed DNA, or sorry, the recombinant DNA, and they've become transformed. So here, if we have any missing colonies, they don't have resistance to tetracycline, so they contain the bacteria that have taken up the recombinant plasmids without the functional tetracycline gene, but they contain the donor gene. So basically what we can do is we can then go to the replica plates, and we know that if we extract some of the bacteria from all of these areas, take them out, they will be our transformed bacteria. So these transformed bacteria will now be able to produce insulin, or whatever the gene was that we've inserted. A little summary shows it here, okay? Now, you can use this little summary to help you basically just give a little structure to this, okay? It's not going into all the details, so we need to add a wee bit more detail in if you'd explain this in an essay or something like that, okay? Now, the last wee step then, the desired protein needs to be extracted, purified, and packaged. But that's basically all of recombinant DNA and bacteria. Thank you.